talk about what's going to be on the up-and-coming exam. That would be a nice thing. So we're going to see if we can get to that point. And in terms of our schedule, so as of right now for us, we have an exam coming, I don't know, in a couple days? Yeah, I think so. So we have an exam on these first few chapters coming up in a couple days. And as per the usual, we have a quiz that occurs 2 days before the exam. So that means by tonight, you guys need to complete quiz number 1. So quiz number 1 is available on Canvas. Remember that it is 20 points. It's all multiple choice questions. So when you're going through it, you want to make sure that you study for it like it's a closed book quiz, but know that it's okay to check your answers as necessary. So it's okay for you to have your lecture notes there, whatever. Go through it, answer all the questions, but before you submit it, double check your answers on any of those that you feel you need to, okay? But it's still timed, so again, you want to be prepared for it. You're not going to be like looking at the question and then like, okay, I'm going to go look that one up. Not going to be like that. We're only 2 days from the actual exam, which is closed book, closed notes, etc. So I think you have 25 minutes and it's 20 multiple choice questions, okay? So you should have some time to go back and double check things, but make sure that you are prepared for it as well. Has to be submitted by midnight tonight. After midnight tonight, you can log back in, you can click on it, and you can review your answers and hopefully the correct answers, which hopefully are very similar. But if there's any that you missed, if there's any that you missed, if there's any that you've missed, then you can see those, and that can help to give you an idea about what you need to study in the next couple days. Okay? Okay. So, we were talking about things like, variation in heart rate or blood flow, like if your coffee spills and you panic about it, your heart rate goes up, and your blood pressure goes up, and all that type of thing. So, one of the things that we were talking about is we were looking at control. Now, we still have some topics to address in terms of this dynamic nature of the blood flow and the blood pressure. We talked about blood pressure and blood flow so far, but we're going to talk also about the capillaries where we're doing that all-important exchange, right? We said that for the most part, our large vessels, our arteries and our veins and our arterioles and our venules, those are about routing the blood from one point to another. The walls are thick enough that we can't get across, okay, for the most part. But it's when we get to the capillaries where we have a single layer of epithelial cells, right, flattened epithelial cells thick, that's where we can do the exchange. So we need to look into the forces behind that exchange. How do we get things to go out of the capillaries into the tissues? How do we reclaim things from the tissues as well? So that's one last topic that we're going to be addressing as well before we finish up today. But before we do that, the last place that we stopped was looking at overall control. How do we control and coordinate the blood pressure and the blood flow, cardiac output from the outside and from the inside? How do we regulate that to make sure that it's matching our demands for the body at the time? Okay? So, what were a couple of things that we talked about last time that were important, do you think? Looking back through your notes or whatever. I don't know what is with the whiteboard markers in here. They must sell them on eBay or something. Hot on the black market.
Talk about blood pressure and the different ways it's controlled.
Yeah, so we did talk about some of the different mechanisms to control it. So for instance, we talked about chemicals that can affect blood pressure, right? So for instance, there are going to be local chemicals that can determine the blood flow into a particular area. It can increase it or decrease it. There are hormones that can work to increase blood flow. Now remember that anytime we're talking about control, and this is a kind of an overarching concept, if you will, for control, this is a big circuit. So things that we modify with the heart affects blood pressure and blood flow. Things that we modify with the blood vessels themselves also affects blood pressure and blood flow. So when we look at things mechanistically, changes in cardiac output, So changes in heart rate or changes in stroke volume, that's going to affect blood pressure and blood flow. But also, if we are, for instance, dilating or constricting blood vessels, that's also going to affect blood pressure and blood flow. So anything that happens along the entire route can affect the local flow, but also affect the pressure and flow overall. Okay? So we talked about some extrinsic controls, including hormonal, but also neural. So with the heart, we have sympathetic and parasympathetic stimulation, which can speed it up and slow it down. It can also make it contract more forcefully or not. And then in terms of the blood vessels, remember that they also are controlled by the autonomic nervous system, but rather than sympathetic and parasympathetic, with the blood vessels, it's just sympathetic. So if we stimulate them with sympathetic neurons, they constrict, and if we stop doing that, then they automatically dilate again. Okay? So that's going to be important in terms of increasing or decreasing heart rate, and as well, the peripheral resistance in terms of the blood pressure and blood flow. Okay, so there's a few different things in there. All right, so where we were stopping last time is we were looking at some of the control that occurs associated with other organs too, and how they have an effect on the cardiovascular system. Yes?
So you were saying this one here.
Yeah.
The heart is controlled by parasympathetic and sympathetic.
Dual control on the heart. Vessels are just sympathetic. Yep, exactly. Very good. So we've seen a few times that the kidneys have a big effect on the blood, right? So we're going to talk about the urinary system later on in the semester and all the details about how it does that. But we know that it's important in terms of monitoring the blood. It's looking at the blood all the time, adjusting it. We know that it's important in terms of creating more blood cells, right? We have erythropoietin that it secretes and tells the red bone marrow to increase the production of red blood cells. But it also monitors the blood concentration and the blood pressure. So it can adjust the blood volume itself, right, by getting rid of fluid. We said that sometimes for hypertension, they'll give you a diuretic, which automatically makes you lose more fluid out of the kidneys. But normally, the kidneys are regulating that. So if you're taking in a lot of fluid, that's a good thing generally, but we don't want to just keep all of that fluid, right? If you're drinking and drinking and drinking, that could cause all of your tissues to become swollen. We don't want that. We want at a homeostatic set point. We want it somewhere where we're maintaining it, not too high, not too low. So the kidneys are constantly monitoring that. They want to maintain a balance in terms of the concentration and the overall volume. So what we find is that there are baroreceptors, right, pressure receptors, that are working on monitoring that. We said that there are various places where they are monitoring that. But through time, sometimes we see that the system can get a little bit, if you will, immune, a little bit kind of used to high levels of blood flow and blood pressure as well. So generally, if we have increased blood pressure, that stimulates the kidneys to eliminate water, and then blood pressure is going to go down, because if we reduce the amount of fluid in the blood vessels, then automatically the pressure is going to go down. There's not as much pressure, not as much substance pushing against the walls. If you have a decreased blood pressure, right, and those baroreceptors sense that, then the kidneys are going to increase blood volume. They're going to work to hold on to any excess fluid that they have to maintain blood volume. They may also hold on to sodium, and we know that if we hold on to sodium, it creates an osmotic gradient, the water follows, and now we're also retaining more water. In terms of those baroreceptors, like we said, in some cases, in most cases, those baroreceptors are consistently monitoring and then sending information back to the central nervous system, and then we can adjust the pressure and the volume accordingly. But through chronic hypertension, when there's too much high pressure, it's interesting because these baroreceptors oftentimes will adapt. So for long periods of time, if they see higher blood pressure, after a while they just figure, okay, well, I guess that's the normal set point. That's where we're going to maintain things. And so they automatically adjust to where that becomes the norm. So rather than being like 120 over 80, it's like, well, whatever, 140 over 100. That's our new norm. So that's going to be our new set point, so we're going to maintain it around that instead, which is not very healthy for the body, right? So what they do is they send signals in, right? They send action potentials into the central nervous system. And so they're always sending some, and if the pressure goes up, they start sending more. Hey, hey, poke, 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 this is getting too high. If it slows down, then their action potential rate also slows down. So when it readjusts to this new rate, then rather than going bing, 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 it's like, whatever, I told you for a while it's going to be high, so now I'm just going to go back to a normal amount of stimulation to the central nervous system and say, okay, I guess this is the new normal. So they've come up with techniques now that are called baroreceptor therapy, where they actually work to stimulate the baroreceptors, in this case the carotid ones, stimulate them, much like a artificial pacemaker stimulates that in terms of regulating the heart rate, and they can stimulate this carotid baroreceptor to increase the amount of signals that are going up to the brain, which then kind of brings that set point back down again. So when the natural way of monitoring these systems gets out of whack, we're finding now more and more extrinsic or external ways to nudge them back to where they should be, back to a homeostatic set point. Okay, so what we find is that the urinary system, the renal, the kidneys, are going to be able to alter blood pressure by altering blood volume. That's the main way that they are involved in that process. Now, they can, by themselves, adjust the amount of reabsorption that they're doing or let it go, but also they can be involved in terms of hormones that can then act as what's called an indirect mechanism, where that affects the blood volume in a different way. So there's this, what's called the RAS mechanism. So the renin, angiotensin aldosterone system or mechanism. This is one where the kidneys will secrete this particular hormone here, and it starts a cascade effect, ultimately stimulating the release of aldosterone. And aldosterone, what it does is it ends up telling the body, and ironically telling the kidneys in a big circle, it comes full circle, tells the kidneys to hold on to more sodium. And like we said, if we hold on to more sodium, just like eating a salty bag of potato chips, it's going to cause the water to follow it. We're going to hold on to water. So this is what's called the indirect mechanism of renal control by utilizing this chemical pathway, this hormonal control, that then ends up allowing us to increase the amount of blood volume, which increases the blood pressure. Okay? So the renal control is another way that we are constantly monitoring and being able to adjust blood pressure and blood volume. So we had neural control doing to the heart and doing to the blood vessels. Now we have the kidneys that have various ways that they can contribute to blood pressure and blood volume. Okay? Okay. So we've talked about all these different ways that we can measure blood pressure internally, right? Our different baroreceptors that are naturally doing it. But of course, we can measure it externally or extrinsically as well. So this is something that we'll do in the second part of the week in lab. We will use an external measurement called a sphygmomanometer. So that's the blood pressure cuff that you are going to put on your patient's arm, and you are going to measure the pressure in an artery, in the brachial artery. So what happens here is this. So you're going to put the cuff on the arm of your patient, and what you're going to do is you're going to increase the pressure in the cuff until it is above, that's what this line is here, until it's above your systolic and diastolic pressure. We know in an artery, the pressure goes up and it goes down. When it goes up here, what's that due to? Why is the pressure going up in an artery?
The ventricles.
Right. The ventricles are contracting, right? So the right ventricle and the left ventricle are contracting at the same time. They're ejecting blood out of the ventricles, 70 milliliters on average, and it's this big tidal wave of blood that enters the pulmonary trunk or the aorta or whatever, right? So that pressure goes up temporarily because there's that much more volume in the vessels. So we have, this is what's called systolic pressure because it has to do with ventricular systole, right, contraction. The bottom point here is called the diastolic pressure, and that's when the ventricles are relaxing and filling. We say that there's no new blood going into the blood vessels, but due to that stretch where the arteries balloon out or pulse out like that, it does an elastic recoil, and so there's still some pressure and blood flow even when the ventricles aren't adding any more fluid into it. So in any case, we see that in the arteries, our pressure is going to vary from high to low, back and forth. So when you're measuring blood pressure, what you need to do is you need to make sure that the pressure in the cuff is higher than both of those. Because temporarily what you're going to do is you're going to compress that artery so that it's closed off like you see there. Okay? So the outer pressure is higher than the inner pressure, so it's going to give way, and that brachial artery is going to temporarily compress. Then you are going to use the valve that we see here. You're going to release the pressure in the cuff, and at some point, now the pressure in the cuff and the pressure inside the blood vessel are going to be equal, and then now, in terms of this kind of war, the pressure in the artery, the systolic pressure, is going to win out, and it's going to start opening up that artery now. And the blood is going to start shooting through that very small diameter. And as it shoots through that very small diameter, it's doing this turbulent kind of movement, much like we were talking about with the heart valves. It's really ejecting through there quickly. It's making a lot of noise as it's going through. Like if you imagine opening the dam, right? Opening the floodgates at a dam, it's really loud when it comes out of there with such force. So as the blood starts now rushing through that narrow diameter of the opening brachial artery, it makes a noise that you can hear with the stethoscope. So those noises that we hear are called the carot cough sounds. And when you first start hearing them, that means you are hearing the systolic pressure starting to win out and open up that blood pressure. So the very first time you hear those carot cough sounds with your stethoscope, that's where you're recording the systolic pressure because the blood vessel is now starting to open up. Okay? Now, as that pressure in the cuff goes down, then the blood vessel itself, the artery itself, is going to start to open more and more. At the point when it opens totally, now the blood's flowing through it very easily in a very smooth laminar flow, and so the noise, that big loud pulsing noise that we heard before, is going to go away. So when we stop hearing those carot cough sounds with the stethoscope, that's where we record the diastolic pressure instead. Okay? So, like the example here, we see, this is something like 120 over, 75 or something like that. Okay? So this is what we're going to be doing in lab this week, and it's just another way in the health sciences that we monitor the cardiovascular activity. So we're doing ECG, we're going to be listening to the heart sounds and the valves using auscultation, but we're also going to be measuring the pressure in those arteries, specifically the brachial artery. Okay? So we know that it's going to vary. We know that overall it should be monitored at a homeostatic level, so it's not too high and not too low. If it's too high, we call that hypertension. If it's too low, we call that hypotension. So systolic pressure below 100 millimeters of mercury, hypotension. Hypertension is when we see those systolic and diastolic pressures out of the norm. So like systolic over 140, diastolic about over 90 or so. Okay? So those numbers can vary too. You can have one be high and the other one be normal. For instance, like if your arteries are getting stiffened, like in atherosclerosis, so the walls are stiffening so that they don't balloon nicely and then have that elastic recoil, then that diastolic pressure number is going to go up because it should have a nice low pressure because we've got this flexibility to it. But if they're getting very rigid, then we're maintaining a higher pressure even when the ventricles are not contracting. Okay? So you can see either of those numbers go up, or they can overall both go up. If we have a lot of blood flow and there's a lot of blood pressure for whatever reason, you might see both of them are extra high. And that, just like the pipes in your house or whatever, if they have too much pressure, puts a lot of stress on them and they can blow out. All right? Okay. So for hypotension, you should be able to define hypotension, and you should know some of these different types of hypotension as well. This is one that probably everybody has experienced. It's called orthostatic hypotension. So in this case, what's happening is maybe you've been laying down and your blood volume has been distributed pretty equally amongst all your different blood vessels, and then you stand up quickly. Now, there's a natural response now to that type of change in position. And what happens is the natural response is you get a lot of vasoconstriction when you do that. So a lot of vasoconstriction along the blood vessels of the lower limbs to make sure that gravity isn't going to cause all of that blood to just pool in your feet. So you constrict those very quickly, and that makes sure that the blood that shunts down there also shunts back out very quickly and goes around the loop still. But if for whatever reason that response is not fast enough or it's not vigorous enough, then, right, Just like, take your water bottle and tip it sideways, you can see that fluid goes this way or that way according to gravity. So now you're up vertical, all the blood that was up here goes down there. And if there's too much down there, not enough up here, then you feel weak and you might pass out, right? So that's orthostatic hypotension. So normally it's not going to happen, but even with that vasoconstriction, say you are dehydrated and you have a low blood volume, right? Even with all that squeezing of the blood vessels down low, if you don't have enough blood volume going around, you're still going to see a lower blood pressure to your head and you might get weak. So there's lots of different things that can come into play there. So orthostatic chronic hypotension, like we said, can come from a number of different sources. If it's always very low, could come from poor nutrition as well as different diseases. Acute hypotension has to do with circulatory shock. When we talk about circulatory shock, it's one of those cases where the common terms that you would use in your everyday life don't exactly match up with the anatomical terms. So, for instance, when you talk about, flexing your muscles or something like that, in 201, where you're talking about actual movement of joints, people talk about, oh, I'm flexing these muscles, I'm at the gym, and I'm flexing all these muscles, right? So, oh, I'm flexing this muscle, and I'm flexing this muscle. Well, in truth, flexion is where you're decreasing the angle of a joint, and extension is where you're increasing the angle of a joint. So when you use the word flex there, it's not anatomically accurate necessarily. And same thing happens with the word shock, right? So we talk about shock, you're like, oh, I went into shock, or this was shocking, I didn't see that coming in terms of the surprise ending of the movie or whatever, right? That's like an emotional shock. That is different from circulatory or cardiovascular shock. By definition, anatomically, circulatory shock has to do with a marked decrease in blood pressure or blood flow so that certain organs are not getting served, right? So when we talk about circulatory shock, that could be due to, for instance, there's what's called hypovolemic shock, where you've lost a lot of blood, and now blood volume and blood pressure can't be maintained. So that's a little bit different. So in this case, with acute hypotension, it's episodic, where there's one point where, for whatever reason, you have low blood pressure, and maybe it has to do, in this case, with circulatory shock. Anaphylactic shock, right? Low blood pressure, and in a particular instance, low blood pressure and low blood flow because your body is overreacting to a peanut or a bee sting or something like that, and it's dilating all these blood vessels, and you open them all up, automatically your blood volume is going to go through, or your blood pressure is going to go through the floor because there's so much now space for the blood to go into. Okay. So in terms of hypertension, you should be able to define hypertension. Difference between primary and secondary, I would say not important for the exam, but it is interesting to know that when we're talking about an increase, a chronic increase in blood pressure, what we find is secondary hypertension is one where they can usually find out what the answer is. So maybe some of our control mechanisms, like in the kidney and the hormones, are influencing it, so it's keeping the blood pressure up. So for instance, if you have a problem with your adrenal glands, there's too much epinephrine going out all the time, or cortisol, stress hormones, it's going to keep your blood pressure elevated all the time. That's not a problem with your heart or your blood vessels, it's a problem with these extrinsic controls instead, okay? So this is from a secondary source, increasing blood pressure through time. Primary or central hypertension, it should be associated with the cardiovascular system, but there's not. The funny thing about that one is that they don't necessarily have, they can't target a very particular root cause for it. So with that one, primary hypertension, usually if you get diagnosed with that, they're like, yep, it's a problem, it's always elevated, but it's probably a combination of these different types of behavioral factors instead. So like stress, smoking, diet, and then just the fact your cardiovascular system kind of gets old and ages. So that is primary or essential hypertension. So that one's kind of harder for them to pinpoint a direct source in terms of the cause. Okay, so blood flow. Like we said, it's key that we are maintaining blood pressure and blood flow throughout the body system. So we make sure all of our different cells are going to be served. We want to get oxygen and nutrients out to it. We want to remove waste products and carbon dioxide from them. We've got to be able to exchange in the pulmonary circuit. We've got to be able to pick up new nutrients in the digestive tracts. And in order for the kidneys to do their job in maintaining homeostasis for blood volume and concentration, we got to make sure that they have adequate blood flow as well. So all of this blood flow is going to be regulated on a systemic level. So overall, we want to make sure it's not too high and not too low. But to individual organs and to individual tissues, it's going to vary as well. Oftentimes in the book, it'll show you like what percentage of the blood is being routed to particular organs. You know that brain has to have a lot, so it has a higher percentage. Kidneys have a lot because not only do they have to be maintained and kept alive with oxygen, nutrients, etc., but they have a job to do on the blood, so they need that much more. So usually brain, kidneys, digestive system, because again, they're adding things into the blood, and then if you have active muscles, they get a larger proportion compared to other types of organs. Okay, so we're maintaining blood pressure and blood flow, and that blood flow can be controlled on a local level, but we always want to make sure that when we're getting our blood out to the organs, we want to make sure that that blood speed, if you will, more velocity, is slowing down so that by the time we get down to those all-important capillaries where the exchange is occurring, it's moving nice and smooth, it's not pulsing anymore, and it's moving very slowly. So we said that in a way, this whole delivery system of blood is kind of like the U.S. Postal Service, right? So it goes far distances very quickly, so you're loading things onto airplanes to go across country or from city to city, or it's going on freeways or something like that. You're not making stops there. You want to go fast from here to there and cover large distances. That's like in the big arteries and veins and things like that. But once you then get to your distribution center, you start getting into the small individual vehicles and going into the neighborhoods, right? Your mailman should not be flying through your neighborhood at like 60 miles an hour. Not only are people going to die, but he or she is not going to be able to deliver to the individual mailboxes and pick up. There's just not enough time before he exits the neighborhood again, right? So when we get down to the capillaries, we want to make sure that it's nice and slow and nice and smooth flowing so that we have adequate time to be able to do that exchange. Not only drop off, but to pick up things from the blood flow. So if we look at this, we start out with the largest artery, which is going to be the aorta, right? We see that down here. Then it's going to branch. So for instance, there's 1, 2, 3 branches off the aorta. What are these? What's the first branch off of the aorta? That's the brachiocephalic artery. Then, left common carotid, left subclavian. So then we see it branching. As it branches, remember as we get to smaller and smaller blood vessels, then the blood is coming into more contact with the wall of the blood vessels, so there's going to be more resistance. So we're going to see it dragging a bit more, slowing down, etc. And so the more branching we have, the more we're going to see the total surface area is going to increase because we're spreading that blood across a greater distance. But we're going to see the velocity, the speed, if you will, the velocity go down further and further. So that's important because as we slow it down and slow it down, we see at the capillaries, it's going to be the slowest. So that's good. So it's going to be low pressure, it's going to be low flow so that we can get what we need done in the capillaries. Then, when we start collecting it in those bigger but fewer venules and bigger and fewer veins, the velocity comes up a little bit. The pressure is still very low, but the velocity comes up a little bit so we can make sure that we can at least get it back to the heart. But it starts out very fast, very forceful, slows down, and then we're going to continue around the other side of the loop and get it home again. Okay, so lots of different control mechanisms, from extrinsic ones, both neural and hormonal, and localized ones as well. This is local regulation. Sometimes it's called autoregulation. And what this means is that if we're going to a particular organ or a particular system, it's going to be localized control. So what happens here is we have chemicals that can adjust the diameter of these blood vessels, but they also can have a little what's called a myogenic stretch, meaning that like the reflex, like the patellar reflex that you did in 201, right? If you hit that patellar ligament, what that does is that stretches your quadriceps muscles when they weren't expecting it, right? So they're sitting there all kind of relaxed. You hit the patellar ligament and they stretch out. And they weren't expecting that, so their automatic response is to contract again and go back to their resting length, right? So it's an automatic response. Same thing in terms of blood vessels. Often, if the blood pressure gets too high, they have this automatic response where they constrict instead. And it happens in the kidneys, and it happens in a lot of different organs. So there's lots of different ways that on a small level, like on a what we call a localized level. We can control the blood flow and the vessel diameter. That's what I was just talking about here, the myogenic control in terms of stretch. There's also all these different chemicals, like we said. If we look at all of these different chemicals and concentrations, that can affect the blood flow and blood diameter. And then there are extrinsic controls like hormones and nerves. So all of these different things here can work to either constrict, like we see it in terms of the green arrows, constrict, or dilate the blood vessels. So we can very, very precisely control the blood pressure and the blood flow overall and to individual tissues and organs, etc. Okay? So this is just showing everything all together in one slide and how those things can have effects. Most of the chemicals that you see at the top there, what they're showing is if we have increases in these, in a lot of these, what it shows is that that tissue is very, very active. And if you have an active tissue, we need to deliver more nutrients and more oxygen to it to keep it going. Okay? Now, in some cases, right, you see a drop in that particular substance, a drop in oxygen, that also indicates that that tissue is really active, like a skeletal muscle, consuming oxygen to make ATP to keep its energy up. Okay? Okay. So, a few different ways. You should know those different ways that we can control blood flow and blood pressure, extrinsic as well as intrinsic, from outside the system as well as inside the system. All right, so we are finally getting down to, like we said, the dynamics of what's going on in terms of the blood flow and the exchange that's occurring out of the capillaries, okay? So we say that it's very important that at the capillaries we're going to have movement go out and movement go back in. We saw that there are different types of capillaries that structurally make it easier or a little bit more difficult for things to get across. But in any capillary, it's about getting things across that wall. That's why the wall is so thin. So we have to see how these different substances that are also important can get across. Now, for some of these substances, which are relatively small, right, like oxygen, carbon dioxide, and some smaller molecules, they can just be moved across in terms of things like diffusion. You may get some facilitated diffusion if you're talking about glucose and things like that. But normally it's just going to diffuse right across the plasma membranes of these cells and go into the surrounding tissues. It might even go through these intracellular gaps, right? So between the epithelial cells, it might go between the gaps as well. So it can go through the cells or it can go between the cells. So that's for a lot of smaller molecules. They just go from where it's high to where it's low. So in arteries, oxygen levels are going to be what? High or low? High, right? Most of our systemic arteries, they look red because they have high oxygen levels. So high in the arteries, high in the arterioles. So when we enter the capillaries, we have a high concentration of oxygen. In the tissues, especially active tissues, we're consuming that oxygen with aerobic respiration, so it's going to be low over there. So there's going to be a natural gradient for oxygen to diffuse from high in the capillaries to low in the tissues. Okay? So for a lot of these things, they'll naturally go the way that they want to go. Carbon dioxide naturally goes the other way. High in the tissues where we're producing it, low in the blood because we've gotten rid of that up the lungs. Okay? Now, if we're talking about fluid and some of the constituents or the solutes in the fluid, that's going to transport in a different way. We have what's called bulk flow that occurs in these capillaries as well that we're going to see coming up. So bulk flow, imagine if you were, kind of transporting lemonade or something like that, and you're pouring lemonade. Often when you pour lemonade, not only are you getting the water, but you're getting the solutes, the actual lemon juice or whatever is in there, or realistically the crystal light that's in there, and you're getting the ice cubes. All of that is flowing together in one big bulk, okay? There is that type of exchange that occurs at the capillaries too. So for fluid exchange, we are not relying necessarily on just figuring diffusion is going to handle it. We have some pressures that are going to work to push or even pull these fluids and their solutes across, okay? So what we find is, at the beginning of a capillary, if we have a capillary, we're going to see this drawn out. If we have a capillary like this, and this is the arterial end, and this is the venous end, right? We know that we're going to have higher pressure on the arterial end because it's closer to the heart, right? We're going to have lower pressure on the venous end because that's further away. We've had that much more drag, okay? So even on the length of a capillary like we see here, it's going to have higher pressure at the beginning of the capillary and lower pressure at the end of the capillary. In general, At the beginning of the capillary, it's about 35 millimeters of mercury. By the end of the capillary, it can be almost half that, 17 millimeters of mercury. We're seeing that blood pressure drop like we keep talking about. So overall, it's relatively low compared to our big blood vessels, but even on the distance of a small capillary, we see it higher at the beginning and lower at the end. So that's going to be important because when we look at what's going on, we said that some molecules are able to diffuse through these flattened cells or diffuse in between them. But when we're talking about natural fluid movements, big volumes of fluid, what happens is it gets pushed out and it gets drawn back in. So at the beginning of the capillary, we have a higher pressure, like we said, like 35 millimeters of mercury. That pressure is fluid pressure. It's called hydrostatic pressure. That's the same as blood pressure. It's just blood is the fluid that we're talking about. Okay? So the capillary hydrostatic pressure, like we said, is high at the beginning of the capillary. Now there's also a pressure that's called the colloid osmotic pressure. Now if we break that down, if we look at osmotic, what does that remind us of? Osmosis, okay? So osmosis is due to the concentration of water in two different areas. Now we're talking about the concentration of water, right? That also has to do with the concentration of things in the water. So for instance, if we had two canisters like this, okay, and this one was pure water, what percent water is pure water? Like what percent? Is it 5%? It's 100%, right? Pure water is nothing else. It's just water. So it's 100% water. Everyone's like, is that a trick question? I don't want to get out there. So if we have something like, I don't know, crystal light or something like that, we add it to the water, now it's not 100% water anymore, right? So now it's whatever. It's like 5% solute, and now it's 95% water, okay? So when we add a solute and we create a solution like that, automatically we decrease the relative amount of water in there. So if we were looking at movement, say between these two chambers, which way would water be moving? It's going to move from high to low. So where's it high? 100%. Over here, right? So water would be moving this way. It would be moving along its gradient from where it's 100% over to here where it's 95%. So there would be osmosis occurring and water would be moving from wherever it's a high concentration to a low concentration. So when we talk about this capillary osmotic pressure, we've got water in the form of plasma inside of the capillaries, but we also have all these other solutes in there too, okay? So the water, in terms of its osmotic gradient, it's looking at inside the capillary and how much of itself is there and how many of these solutes are there, and it's looking on the other side in the interstitial space, in the tissue. And it's saying, okay, how much water is over there? If it's lower over there, I'm going to move out. If it's higher over there, it's actually going to come into the capillary instead. Okay? So there's osmotic pressure inside of the capillary, and there's osmotic pressure technically outside the capillary as well. So these are various forces that are working to push water one way or the other. One is a fluid pressure, like we see here. The other one is an osmotic pressure. So let's take a look. I want to show you a figure that is going to try to put this all together, okay? So net filtration pressure, this overall, don't worry about this whole calculation here, okay? Net filtration pressure overall is looking at all these forces acting in the capillary bed. So we want to look at this one right here, okay? Okay, so here's the arterial end of the capillary like we said, and we said that at the beginning of the capillary compared to the end of it, the hydrostatic pressure is going to be higher. So about 35 millimeters of mercury at the beginning and about 17 millimeters of mercury at the end. Okay? This is due to hydrostatic pressure. When the blood gets to the capillary, it's got two choices, right? It's like exiting, Chase Field or something like that after a game. All these people are trying to go through these few exits, and it's a big flow through the small area, right? So you have two choices when you're exiting. You either go with the flow because there's always more behind you, or you jump out to the side and wait for your friends, right? And let the flow go past you. So the same thing in terms of fluid that's showing up in the capillaries. There's more fluid always showing up behind it. So either you shoot straight through the capillary or you can jump out to the side. And if we're jumping out of the capillary, we're going into the tissues. So that's what water and some of its solutes can do. They get pushed out. So that hydrostatic pressure tends to push water out of the capillaries at the beginning of that capillary bed there. So we see this net pressure out right here is due to mainly the fact that we have a high amount of hydrostatic pressure pushing this water out to the side. Now the osmotic pressure that we see next to it, osmotic pressure, like I said, has to do with water as well as solutes. Remember that when we were talking about the plasma way back when, this is a little bit of a review, right? We talked about blood just as a tissue. We're going to look at the cells. We need to know all about the cells. What are the different cell types and what do they do? But we also need to know about the plasma. What do we find in the plasma? We find hormones, we find different ions, there's nutrients and waste products, but there's also plasma proteins. Refer back to that table where it lists all the different things we find in the plasma. There's 3 different plasma proteins that we commonly find in the blood. One of them is fibrin, technically fibrinogen, right? What's the importance of fibrin or fibrinogen? Clotting cascade, right? So in its inactive form, it's soluble, it's floating around in the plasma, just waiting to become activated and become fibrin threads to reinforce the clot. So that's one of our plasma proteins that's floating around all the time. Second one are called immunoglobulins. Those are antibodies. So antibodies, like we learned about with red blood cells, right? Antibodies are proteins floating around in the blood looking to attach to a cell or some sort of a particle that shouldn't be there. Okay, like a red blood cell that got in there or a bacterium or something like that, okay? The third plasma protein that we have in there is called albumin. Albumin's main role is to create an osmotic pressure. So if we have these albumins floating around in the plasma, that automatically kind of creates a suction because what's happening is we got a bunch of plasma proteins. That means we have relatively less water in the plasma. And so that creates a gradient for the water that's working to draw fluids back into the capillary, okay? So the whole role of those albumin proteins that are floating around is to create an osmotic gradient and suck water back into the capillaries. So that's why you see that little arrow right there of 25 millimeters of mercury. This arrow is due to the osmotic pressure inside of the capillaries trying to draw that fluid back in. So look at these 2 arrows though. These arrows are going opposite ways. So we have 35 millimeters of mercury that's trying to push out. We have 25 millimeters of mercury that's trying to push back in. If we look at those together, obviously in terms of a tug of war, in terms of pushing this way and that, the 35 is going to be bigger. So the net filtration pressure at the beginning of the capillary, between those 2 forces, the hydrostatic pressure is high enough that it's winning out and we're getting a net movement of fluids out of the capillaries at the beginning, which is exactly what we want. We want things to be delivered to the tissues at the beginning, okay? But by the end of the capillary, we want to make sure that we're drawing things back in. We can't just constantly push things into the tissues. Our tissues would just swell up and we'd have edema, right? We want to exchange. So by the end of the capillary, we want to be able to draw fluids back in, which we see here. Now that osmotic pressure that we mentioned, that's going to be the same, right? We still have the same albumins in there working to try to draw water back in, create an osmotic gradient. But we know that the hydrostatic pressure is going down and down and down and down due to that drag. So by the time we get to the end of the capillary, where it once started at about 35 millimeters of mercury, now that hydrostatic pressure is weaker and weaker and weaker. So by the end of the capillary, you can see it's down to like 17. So when we look at those 2 forces that are pushing this way and that by the end of the capillary, the hydrostatic pressure has become weakened. Now the osmotic pressure is strong enough, it's winning that battle. And so now the fluid starts to be drawn back into the capillary by the end of it. Okay? So the point here is that for the exchange of fluids, like we talked about, this whole idea of bulk flow, there's 2 main forces involved. The force that pushes it out is the blood hydrostatic pressure, and that's occurring at the beginning of the capillary. The force that's drawing it back in is the osmotic, the blood osmotic pressure, which is drawing it back in by the end of the capillary. Okay? So you need to know those 2 different forces. You need to know that hydrostatic is stronger, and that's winning at the beginning. And you know that osmotic is going to be, the blood osmotic is going to be stronger and winning by the end, drawing it back in. Okay? I know that's always kind of a tough topic because it's down at a microscopic level, and we're talking about pressures this way and that way, etc. But that's important in terms of getting these things to move across into the tissues and out. Okay? So I don't particularly care about the actual numbers, but I do care about the trend. Where are the two pressures high and where is it low? Where is the movement occurring out and where is it occurring back in? Okay? So questions on bulk flow and this net filtration pressure? Yeah?
Just wondering if that whole process, hydrostatic pressure and osmotic pressure, that whole process is called bulk flow?
It's called bulk flow, yeah. So this process of exchange is called bulk flow. The movement of large amounts, relatively large amounts of water and some of those solutes, pushing them into the tissues and reclaiming them. That is bulk flow.
Okay, net filtration.
Net filtration pressure is the calculation that you can do to say, okay, if we take all these forces together, going this way and that way, what's the net? Like when you talk about your paycheck, right? There's a big difference between gross and net, right? Net is when you look at all those things adding in, taking out, what do we actually get at the end of it? That's what we're seeing there. Yeah. Okay. Everybody all right with that? All right. So as we get towards the end here, like we mentioned, we do have some, kind of, like I said, we will at times address particular diseases and disorders that may be relevant to that particular system. Now, cardiovascular system is a big one in terms of the health professions, right? When you are taking your vitals on your patient and when you're monitoring things, this system, because it has this delicate relationship with all the other systems, we want to make sure that this system itself is working properly. And you want to be looking at, the content of the blood, so they're doing blood samples, making sure that the pH is right, making sure blood volume is right, etc. So we have to monitor these things, even to the point, when we talk about this exchange here, this whole balance is when it is homeostatically controlled and everything's normal. But these things can get out of control. Right before this, we were talking about chronic high blood pressure, hypertension, right? If your overall blood pressure is chronically higher than it should be, then at the beginning of this capillary, it's not going to be 35. It could be like 40, 45, 50, 55, something like that, right? And if you have that much more pressure at the beginning of the capillary, there's that much more force pushing the fluid out into the tissues. So people that have chronic high blood pressure or even episodic high blood pressure, they can get swollen out in their tissues because this fluid is being pushed out of their capillaries and it's ending up in the tissues. So it's not in balance, it's not an even exchange anymore. We have too much pressure pushing it out, and by the end of the capillary, when we look at that balance, maybe that hydrostatic pressure is still winning a bit. Now osmotic pressure isn't able to draw it back in as effectively either. So you get swollen like that, okay? So osmotic pressure, that can be affected too. So if we don't have enough plasma protein, so if the liver isn't doing its job, we're not producing these different proteins, then there's not enough suction pressure to draw these things back in. So again, you can become swollen out in your tissues. So they're monitoring these things to try to see what's going on in the body. Because everything circulates around, you can take one blood sample, or you can take one measurement, and you can get indications about what's going on in a lot of different organs and tissues. Okay, so as we mentioned, in terms of diseases and disorders, for shock, you should know what shock is, what cardiovascular or circulatory shock is. So like we said, this is a condition when we don't have enough blood pressure or blood flow for the blood to be able to circulate normally. So if you're not getting enough delivery to your tissues, they're going to start to suffer, particularly your very active tissues like your brain, your heart, etc. Okay? So there are three types of shock. This one, hypovolemic shock, means low volume, right? So if you have been out swimming and you're surfing and your leg gets bitten off by a shark, doesn't often happen, right? You have more of a chance of getting hit by lightning than that happening, so don't panic. But if it did happen, then what happens is you're losing a whole lot of blood, right? And so as your blood volume goes down, even though we have these responses like your heart rate's going to go up to try to keep the pressure up, and your blood vessels are going to constrict, etc., doesn't matter. Your blood volume is dropping so fast that you're not going to be able to keep blood pressure up. And so that's called hypovolemic shock, okay? There's also vascular shock, and vascular shock is where the blood vessels themselves are responding in an extreme way that's dropping the blood flow and blood pressure. So this is oftentimes when you think of anaphylactic shock, right, with an extreme allergic reaction. This is an example of vascular shock. So it's the blood vessels themselves. Usually when we're talking about the immune system and defending, right, if you get a bite or something like that, you get localized blood flow. You get redness and swelling at the site. Because we're like, I don't know what that is, but we need our defenses there. So let's get some red blood cells in there to add in oxygen to that tissue or whatever. Let's get some white blood cells in there to attack whatever that is. So you get localized swelling. But in the case of an extreme allergic reaction, you've got this allergen that's circulating around, and your system is so panicked about it, it's like, I don't even know where it is, so we're just gonna put everything on alarm. So instead of just dilating, where it came in, right? So if you have like low-level allergies, like pollen or ragweed or something like that, where do you get your swelling? Right there, right? This is where your allergen is attacking you, in your respiratory membranes, your mucous membranes, etc. So that's where you get your swelling to try to attack it. But anaphylactic shock, it's just, it doesn't know what to do, so it just opens up everything. And when you open up all these blood vessels, now the blood pressure is going to drop. So it's going to drop significantly, and you may not be getting adequate blood flow to your different organs. So that's why, right, people that have the possibility of anaphylactic shock, extreme allergic reactions, what do they carry with them? EpiPen, right? EpiPen at $600 a pop or whatever, that's in the news these days, right? The main producer of EpiPens have a really high price on it. But the EpiPen has epinephrine in it. We know that with epinephrine, when you inject that, then it's going to increase contractile, right, stroke volume as well as heart rate. So that's going to increase blood pressure. It's also going to act as a vasoconstrictor. So that's going to work to increase blood pressure as well. So it's going to counteract those effects. Okay, last one is, this is a problem with the blood. This is a problem with the blood vessels. This is a problem with the heart. Right? All of these are connected. Any one of those three things goes out of whack, the whole system gets affected. So a cardiogenic shock. So for some reason, maybe you have an enlarged heart or something like that, or you've had a heart attack and the muscle cells are not able to contract properly. So in that case, right, if you are in ventricular fibrillation and you have all these spasms going rather than nice smooth contractions and refilling, then you're not going to be getting a good amount of volume going out to the system. So any problem with the heart itself can also lead to this circulatory shock. So that's called cardiogenic. It is generated from the heart, a problem with the heart instead. Okay? Okay. So the end of this chapter really just goes through a little bit more of the anatomy, stuff that we've been doing in lab already, right? So the different circulations, this has two. Technically there's three though, right? So there's pulmonary, there's systemic, and then what's the third one? Coronary or cardiac, right? That mini circulation that's going around the heart itself. But the rest of this is really just looking at some of these different systems and the distribution of the blood throughout the system. Okay? Now, I'm not going to go through these different arteries and veins. These are things that we're studying. We've got lots of time to study that coming up to the lab practical next week. But I will review for you these differences because I like this figure in comparing the fetal circulation with the adult circulation. Because the fetal circulation, it's set up anatomically different because we have a whole other organ that is contributing to the exchange of nutrients and waste products and respiratory gases, etc. So it's not just to and from the lungs for respiratory gases, to and from the intestine for nutrients and waste products and things like that. We have this whole other organ called the placenta where we're going to route blood vessels out to and from where we're going to be doing those exchanges. Okay? So in the fetal circulation seen here on the left, notice that that whole separation between arterial high oxygen blood and venous low oxygen blood is kind of messed up, right? Luckily, the fetus is not very big and not doing a whole lot of activity necessarily, and so this is still enough oxygen supplying and nutrient supplying for the tissues that we can mix them and not have it be a problem. But here's that placenta, right? So we need to be able to get our kind of waste accumulated blood and carbon dioxide accumulated blood out to it. So we have, coming off here, off of the internal iliac vein, we have what are called umbilical arteries, and there are two of them. So the umbilical arteries are going away from the fetal heart, remember? Right? That's why they're arteries. They go into the umbilicus, and they go out here to the placenta. And as it hits the placenta, we get rid of waste products, carbon dioxide, we pick up oxygen and nutrients, and now we're on our way back. We also pick up fluid there, right? It's one of the reasons that the maternal circulation, the blood volume goes up, the levels of nutrients and sodium and all those types of things go up in the maternal circulation, because not only are we supplying the woman's body, but we're supplying it to the fetal circulation as well, right? It's not eating and drinking, so it's got to be able to get even things like fluids and nutrients from there as well. Okay, so umbilical arteries taken out. There's one umbilical vein that goes back the other way. So the umbilical vein is going to have this oxygenated blood, nutrient-laden blood, going back, and we see it here, okay? Then it connects to this specialized connection, this bypass there, which is called the ductus venosus. So we said that that name suggests that it is a vein also returning blood back to the heart, okay? Dumps it into the inferior vena cava, goes into the right atrium. Now we kind of have a mix, right? Because the inferior vena cava has deoxygenated blood coming from the body and highly oxygenated blood coming from the ductus venosus. That's why you're seeing it purple in the heart there, okay? Right atrium, from there we normally go right ventricle, then to the pulmonary circuit. But we've already done our oxygen exchange out here. So why are we going to send it all the way to the lungs again? We're going to send some to the lungs to keep them alive, right, like organs, but we're going to shunt it through the shortcut right here, right, through the foraminal valley from the right atrium to the left atrium, and then we can quicker get it out to the systemic circuit that way. We know also there's a second shortcut right here, which is going to be the ductus arteriosus. That is also going to help to get this already oxygenated blood with the nutrients in it to the systemic circuit that much faster and out to the body like we see there. Okay? Now, these different functional vessels and shortcuts in the fetal circulation basically seal over and dry up in the adult circulation, right? We have the ligamentum arteriosum, which is going to be up here, okay? That little white connective tissue. We have down here the ligamentum venosum, which dries up and is next to the liver. And down here, you have what are called the umbilical ligaments, which are former umbilical arteries, as well as you talk about that umbilical vein, you see that drying up. So there are still physical connective tissue connections between these different places. This becomes fossovallus, but there's no blood flow going through there, or there shouldn't be. So later on, like when you're studying or doing your quiz, you can test that out, see that there are physical connections between your umbilicus, your belly button, and some of these internal organs. So if you were to take your finger and put it in your belly button and move it around significantly, you will actually feel a pull inside as these umbilical ligaments that we see here are still connected to some of those deep blood vessels, right? So luckily you can't, be whipping it this way and that way, but even subtle movements on the order of a centimeter here will pull on those ligaments and you can feel the pull on the inside. Yes. Wait until you're in private. Don't be walking through a hallway, right? You'll think you're a little bit odd. Okay, so this just goes through, like you said, those fetal bypasses and adaptations to that circulation, talks about what they're doing, okay? All right, so that takes us to the end of that chapter on blood vessels and hemodynamics. So, when we're thinking about the exam coming up, exam format, first and foremost, we're going to have mostly multiple choice questions, multiple guess questions, right? So the answer will be there, but realize that on some level, right, that seems easy, where you're like, I don't have to study that much. The answer's right in front of me. But there's always those levels of kind of how difficult those answers are, right? First couple are like, oh, that's totally wrong. That's very obvious. The last two is like, you're like, I knew it to that level to cross out those ones, but now I'm not sure about this level of detail. Okay, so make sure that you're still studying, even though we're going to be mostly multiple choice questions, that you're spending enough time learning the detail of these major topics that we're going to be talking about, or have talked about and we'll review. Mostly multiple choice questions. We're going to have commonly a vocabulary section with the vocabulary words you have in the exam objectives and study materials module. You've got a list of the vocabulary terms that you need to know for each exam, including this one. So you need to make sure you go through those. There are exercises to help you study those as well. For those, you'll get questions in either one or two formats. Either we'll give you the sentence and there'll be a blank in the center where you've got to enter in your vocabulary term, or it'll just list the vocabulary term and you are writing in your own definition for it. It's kind of like one or the other. Now realize, in either cases with those, first question you get, do we have a word bank? You as individuals, yes, you have a word bank. Word bank. So throughout the last few weeks, you have been making deposits on a regular basis, and on exam day, you are going to make withdrawals from the word bank. Okay? So when you get to your careers, which is what your whole path is here, trust me, no word bank. So you've got to have these terms in there. Okay? So it'll either be, like we said, you're putting in the word, and I give you the definition of the sentence, or I give you the word and you give me your definition. So there's kind of pros and cons to each. If you give the definition, there's a little bit more flexibility in terms of exactly how you describe it, and it can still be different but okay between two different people. And the other way, I'm giving you the sentence and you have to recognize that that accurately describes this particular vocabulary term. Okay? So a small section of that, maybe no more than 10 points of that usually. Okay? Then there is usually a short answer section. And a short answer section is where we are asking you oftentimes to compare and contrast things or describe how these things work. So it's usually a more complex process where, like with a ECG output, right? Being able to draw that out, label the waves, and describe what's going on during each of them, right? You can't just do all that in a multiple choice question. So that takes a number of points. We need to give you some space to do that. So sometimes they're drawing questions. Sometimes you will see they might be what we call tracing questions. Like it'll say, trace a molecule of oxygen from the time it enters the heart until it arrives at the lungs. So it could then start, with that right atrium and going through the valve, past the valve, into the right ventricle, through the pulmonary valve, to the pulmonary trunk, to the pulmonary arteries, and then out to the lungs, right? So those tracing questions are really generally more anatomy questions, but it's making sure that you understand how these things are flowing through, okay? We could do compare and contrast questions where we're like, we really looked at the structure and the function of these different blood vessels. We have all these different types of blood vessels. Arteries, arterioles, capillaries, venules, veins. We even have 2 different types of artery. We have 3 different types of capillary. Tons of different blood vessels. Each one, their anatomy is a little bit different, and it has to do with their particular function. Letting things through easier, restricting blood flow and controlling it, whatever, okay? So in that way, it could be, name 4 types of blood vessels, and then compare their anatomy and physiology, or compare their structure, talk about their specialized structure, and how that relates to their function. Now, if I say something like 4, we just said there's like 10 different ones if you include the different capillary types and artery types, etc. So if I'm doing a compare and contrast, oftentimes I'm giving you options about what to tell me, right? So if I say 4, you can pick any 4 you want. The ones that pop into your head, your favorite ones, whatever, okay? So that makes it a little bit easier in terms of these short answer questions as well. Usually you've got some options in terms of your answer. Other types of things. Like we said, for the ECG, sometimes these short answer questions will be drawing questions. So remember, we spent a decent amount of time talking about how the cardiac muscle cells, you have these 2 populations. There's autorhythmic ones and there's contractile ones. They're located in 2 different places in the heart. They are different in terms of their membrane potential changes. So, drawing out the membrane potential, those little curves where it kind of goes back and forth, or does that kind of cursive R type of thing. Being able to talk about that and say, this is how a contractile cell works, and this is how a conductive cell works, or whatever. That's another type of compare and contrast. Okay? So we start out with blood. Blood, we talked about cellular elements, fluid components, and then we talked mainly about the clotting cascades, right? Extrinsic and intrinsic. You don't need to know all the factors. You should know, where does extrinsic occur? Where does intrinsic occur? What starts them off, this whole ball rolling? In terms of the chemical cascade, intrinsic is the full set of all the different clotting factors. The extrinsic is kind of a shortcut. It doesn't involve all of them, so it's a bit faster. But ultimately, the end of those is where we have fibrinogen becoming activated to become fibrin and reinforcing that platelet plug, okay? Now, before that, remember the first 3 steps to what we call hemostasis is, first step is vascular spasm, where the cut blood vessel is like a fire hose and it's trying to kind of constrict itself so it's not flowing all over the place. Second one is the formation of the platelet plug, where those thrombocytes, and you should know the fancy name in addition to the common name, the thrombocytes or the platelets are starting to stick to the collagen fibers and stick to the broken blood vessel, and they're starting to stick to each other. But then we need to reinforce them to form an actual solid clot. That's the third step, is clot formation, the whole extrinsic and intrinsic pathways, etc. Okay? Another major topic in the blood chapter, we talked about blood typing, right? So you need to know the different blood types. What does it mean in terms of antibodies and antigens, etc. Rh factor as well as ABO. Anybody think of anything else in terms of blood? Or have questions about anything in blood? Yeah.
Immature blood cells and.
Yeah, good point.
cells.
So you should also have an idea of, we have this whole hemostasis in terms of, we want to maintain homeostatically, you can say correct or basically the right amount of red blood cells, of white blood cells, and platelets. And each one of those is controlled independently, right? So for instance, with red blood cells, it's the erythropoietin, the EPO, which is a hormone from the kidneys, which tells the red bone marrow to produce more red blood cells. So we should know that the red bone marrow is where all of these cells come from. That's important. And then you should know the controlling factors as to what controls when these blood cells are produced. Erythropoietin for the red blood cells. For the white blood cells, we have these different chemicals that are called cytokines. It's a category, but we have different ones for eosinophils or neutrophils or monocytes. We just say cytokines are the ones that specifically will target each one of those in order to increase their production. And then for the thrombocytes, we have thrombopoietin instead to make sure our platelet levels are right. Now, in terms of developing, they all come from the red bone marrow, right? But they have these early intermediate stages. So we start out with this very, very generic pluripotent stem cell, right? It looks like a very generic cell. It doesn't look like any of the particular red blood cells or white blood cells, etc. We start out with a pluripotent red blood cell. And then that one is basically like a cytoblast that then is going to give rise to all these other cells. We have 2 different cell lines, okay? We have myeloid stem cells and then lymphoid stem cells. Myeloid stem cells give rise to red blood cells and all 4 of the white blood cell types except for the lymphocytes. Lymphoid stem cells give rise to the lymphocytes, okay? So you should know those precursors, the myeloid stem cell line and the lymphoid stem cell line. For the myeloid stem cell line, when we're looking at, okay, producing red blood cells, there is a stage before the last mature red blood cell, which is called, right, the erythrocyte. There is one stage before that, it's called the reticulocyte. Now there's a few other stages before that, but the one stage before that you should know is the reticulocyte. That's the last little immature stage cell. That's where it kind of still looks like a cell with a nucleus and organelles and stuff like that. But at that stage, it's going to start ejecting those and take the final form of this bag of hemoglobin. That's another important topic, right? You need to know what hemoglobin is. You need to know that hemoglobin is the protein that binds the oxygen, has 4 binding pockets to them, have iron at the core of each of those. It can bind oxygen. It can bind carbon dioxide, actually, as well. So you need to know about hemoglobin. So reticulocytes you should know as well. There's also, in terms of the generation of our platelets, is this massive cell that's called a megakaryoblast, right? That's the precursor cell before we get our thrombocytes floating around. This massive cell that kind of gets shredded like cheese as it exits the red blood cell and creates all of these fragments that we call platelets, okay? So yeah, those are good topics for blood also. Heart. So remember that between the lecture exams and the lab exams, lecture is going to tend to focus a little bit more on physiology. Lab is going to tend to focus a little bit more on anatomy, right? What's this, what's that, what's that? Here, you may get some of that, right? So you may get maybe 1 or 2 diagrams where it's like, what is this particular structure of the conduction system? This is the SA node. Or what's this particular layer of the heart that it's pointing to? And that's the epicardium or something like that. But but those would be rare on the lecture exam. We're talking more, how do these things work? We got a lot of detail in terms of blood clotting cascades or whatever. We want to focus on those things as opposed to what's this, what's that. Okay? So when we get to the heart, sure, there can be some in terms of anatomy and like the different chambers and different valves and stuff like that, but it's more getting into the details, like we said, talking about those different types of cardiac muscle cells, talking about this whole thing in terms of the blood pressure and cardiac output and how that's adjusted in terms of the contraction or the stroke volume and what's working with that and what's working against that. Are you talking about preload and postload and stuff like that or whatever? And then also that whole heart rate, what determines heart rate, etc., the function of the heart. So we talked about that in terms of heart, then we got into blood vessels, same type of thing. A little bit of anatomy, looking at all these different blood vessels and how they are structurally supporting their function. But then we had a whole section, the second half of that lecture for that chapter is more about physiology. It's about blood pressure and blood flow, ways that we can adjust blood pressure, ways that we can adjust blood flow. Talking about exchange of the capillaries, that bulk flow is always a big topic in terms of knowing those two major pressures, hydrostatic pressure, pushing it out, the osmotic pressure, drawing it back in, etc. Other things, heart, blood vessels. Yeah?
There's a question about tracing blood through the coronary circulation.
Through the coronary circulation?
Do we
So-
know that or just like
That's a good question. So for tracing questions at this point, we would probably focus on through the heart as opposed to through the vessels serving the heart. Given that your lab practical is like a whole other week, I mean, you've done heart, you've done blood vessels already, so technically you would be ready for a tracing question, like trace a drop of blood from the front side of the calf or the front side of the leg until it gets back to the heart itself. You could do ones like that, but I would say, given the fact that the lab practical is still a ways off, we might restrict it to flow through the heart, like we said, through the chambers, but coronary circulation, tracing questions, I wouldn't worry too much about that. I mean, you know where those blood vessels are, so you know it's an artery, it's going away. You can see this one's on the front, that one's on the left side, this one's on the right side. It would be general like that, but I wouldn't worry about that. Other questions? Dead silence is a no. Good to see you're consistent. All right. So what that means is we've got a quiz tonight for sure. You have to do calculations for cardiac output. You should know what how to calculate cardiac output in terms of stroke volume times our heart rate, but there won't be any calculations in terms of actual numbers for that. I wouldn't worry about that. Okay? All right, so make sure you get your quiz done by midnight tonight. Review your quiz after that. If you need help, study lab, office hours, come see me. Otherwise, next time we see each other, we'll be doing a little exam.